Hi everybody, and welcome to the second part of the cells. So we're going to be continuing on to technically the working cell. So last week we learned about the different parts of the cells and the differences between animal cells and plant cells and eukaryotic cells and prokaryotic cells. Remember, prokaryotic cells are bacteria, not plants. So keep that in mind when you guys are going through all of these different lectures and stuff and potential test questions that I could totally ask you guys. So today we're going to be working on the working cell. And this is essentially what the cell is actually doing. So we kind of learned about the different types of cells and some of the different organelles. And now we're going to be talking about specifically what are they actually doing and why are they doing the things that they're doing. Okay? So let's go ahead and get started. First off, we need to define energy, because when we're going to be talking about this chapter is cell energy. And so remember, we kind of learned when we were talking about macromolecules that, you need that cheeseburger to survive because you need the proteins and the amino acids and the lipids and the carbs. Well, you need those things because your cells need energy. And the only way the cells can get their energy is if we ingest food. We're not plants. We can't just make our own energy, right? We need to ingest our own food to actually be able to get that energy we need. So what is energy, right? We keep talking about energy and I need energy. Why do I need energy? What is energy? Energy is technically the ability to cause change. Okay, so if you can cause change, that is energy. So if you think about, I want to push something over, right? That's going to require energy because I'm changing it, I'm pushing it over. Now it's changing position. So that would be like the change that we're talking about. If we're talking about a chemical reaction, maybe you're changing something from two simple monomers into a more complicated polymer. Right? That's a chemical reaction that's changing, that requires energy or the ability to cause change. The capacity to cause change, same thing, ability. When we talk about kinetic energy, right? If you've ever known anyone who's studying kinesiology, that's the study of motion. So kinetic energy is the energy in motion. If I were to take this little remote control and chuck it across the room, I've added energy to it. Now, as it flies through the air, it has kinetic energy. Once it would, say, hit my kitchen floor, then that energy would be transferred to the kitchen floor and it would probably cause it to explode because that's a transfer of energy. Remember, energy is never created nor destroyed, it's just moved around. Okay, so if I didn't throw this across the room, say if I just left it right here, right, it also has what's called potential energy, or the energy that it could have, right, because it could change. So this location right here is several feet above my floor. So all we need to do is push it, give it a little kinetic energy, and now it's falling, right? So it goes from that potential energy, the sitting energy, to that moving energy, the kinetic energy. So again, we're just transferring this energy. We're kind of moving it around. Now, if that confuses you a little bit, this is a diagram of kind of hopefully to help you kind of solidify that information. Now, if I am all the way down here and say in the water, you don't have a lot of potential energy because where else are you going to go? Well, you're not going to naturally go anywhere. You would have to add energy and climb up that ladder. Okay, so your potential energy is really, really low. What change can you really cause? When you're in the water, not much. You would have to add that energy to be able to crawl up the ladder. Now, if you're standing at the top of the dive board, then you have high potential energy because where could you go? You go off that diving board, right? Really easy to fall down, kind of hard to go up. Right? Same kind of thing when it comes to kinetic and potential energy. While they are jumping in the air and they are falling down, that is kinetic energy, the energy in motion, right? But they had a lot of potential energy when they were up here on the top because all they'd have to do is step off and now it's kinetic energy. So again, the potential energy is kind of like the energy that you could have. When we talk about all these bonds and stuff that we're making our macromolecules out of, right? You're breaking and making these bonds, you're breaking and making these bonds. That's energy. That's in fact potential energy. Because as you stored that, as you created that bond, you could break the bond and release the energy, right? But you haven't yet, which is why it's potential energy. It's still stored in this bond right here. And only once you break that bond are you going to release that energy, releasing the potential energy, the energy that could cause change. So hopefully that kind of makes sense to you guys. If not, luckily for you, this is about as much detail as we're going to actually go into the subject. So just keep things in mind. That big cheeseburger, a lot of potential energy, right? Because if you break up all those bonds, you release said potential energy. And you have that energy now to use and utilize. So that's why it's potential energy. It's energy that we could be using. It's sitting there. It's waiting. It's like, I'm ready, right? Guy standing on top of the diving board. I'm ready. And then he jumps off and it becomes kinetic energy. That kind of thing. All right. So again, when we're going through these operations, these chemical reactions and are inside of our body, right, it kind of goes back and forth between kinetic energy, potential energy, kinetic energy, potential energy. Remember, energy is not created nor destroyed. It's just moved. So this is kind of how we move it. We're transferring it from one source to another, et cetera, et cetera, et cetera. And sometimes this transfer is really, really important because what you're doing is you're turning on and then turning on and then turning on and then turning on and turning on, which causes a bigger reaction, right? These little tiny mechanisms just getting switched on are actually going to cause things like, I don't know, us to live. Yeah, I'd kind of like to live, wouldn't you? So keep that in mind when you guys are going over this potential energy, kinetic energy. It's just, again, there's different forms of the same energy, the potential to cause change. All right. Now, we talked about this whole energy can't be created nor destroyed, and you're like, wait, what? It's called the law of conservation of energy. Conservation, we're conserving the energy because the energy's not going anywhere. It's just getting transferred around. So this is one of Newton's laws, right? And it is true, right? That energy does get passed forward. If I took the remote and chucked it across the room again, that energy would get transferred into the tile and it would form an eruption of an explosion of my remote being blown up because that energy has to go somewhere. It doesn't just stop. All right. So just converting energy from one form to the other, typically kinetic to potential, potential to kinetic, vice versa, back and forth. All right, so usually what's happening in our body is not every last little bit of energy is used, and a lot of that is actually given off as what's known as metabolic heat. So if I eat that cheeseburger, yes, I'm going to get a bunch of energy out of it, but I'm not going to get every single last little bit of energy out of it, right? That's just kind of, a lot of it's going to be lost as it passes through our digestive system. That is what it is. It's leftover energy, essentially. And then the rest of it is going to get pretty much just evaporated out of us via this metabolic heat. So as I digest stuff, I create heat. That heat is a form of energy. So instead of me using it, it actually gets lost and, well, not lost, transferred to the environment as metabolic heat. So we're actually causing a lot of energy to be released out of our system just by being alive. Just by being alive, we're actually going to release that heat. That's why we give off heat. Yeah, crazy. All right. So again, that heat is just kind of a, it's a measure, heat and entropy are kind of the same thing. It's just a measurement of heat, this metabolic heat that we're giving off, this chemical energy that we're giving off. But again, don't worry yourself too much about that. This is not a chemistry class. We're just kind of, why do we need energy? Who needs energy? What kind of energy are you talking about? That's kind of where we're going with this right here. So again, just smaller, big picture stuff. That makes sense. All right, so let's talk about the energy that our cells need. This energy molecule is going to be known as ATP, or adenosine triphosphate. Triphosphate because there's 3 phosphates. So that's what we see right here. This is the adenosine or adenosine, however you want to pronounce it, it's totally up to you. But this is the 3 phosphates, that triphosphate that we're talking about. 1, 2, 3 phosphates. Now, when we talked about our enzymes, remember we talked about that enzyme being specific to one substrate, right? It goes and picks up that one thing. If we're talking about lactase, right? It's going to pick up lactose. That's the only thing that fits here. And then it's going to drop off its lactose and it's going to get recycled. So the enzymes are always recycled, okay, to pick up another piece of whatever substrate they're going to chemically react and then move it on. So essentially they come in, they pick up that substrate temporarily, take it where it needs to go, drop it off. The enzyme then goes to pick up another substrate, right? And then drops it off where it needs to go, and then goes to get another one. That is kind of like this molecule here, adenosine diphosphate. So you can kind of think of it as like a temporary energy carrier, right? So it's going to pick up this 3rd phosphate molecule, and then it becomes ATP, adenosine triphosphate. That's energy, right? That's cellular energy. So it's kind of just like, all right, I'm just kicking it until I pick up that extra. Oh, I've got the extra phosphate now. So when you create that bond, remember, you're storing energy. Okay, this is now potential energy. So this is what makes it the high energy molecule that we know as ATP. So this ATP molecule, this high energy molecule, is energy, right? That's how we use cellular energy. So it forms a bond. Now it's stored that energy. It's going to take it where it needs to go. Then it's going to release that phosphate. It's going to break the bond. And then that phosphate is going to go off to live its little phosphate life. And then this diphosphate, the adenosine diphosphate, or ADP, is going to go pick up another phosphate. Same thing like the enzyme. Picks it up, takes it where it needs to go, drops it off. In this case, it's dropping off a phosphate. So then it's going to go back, pick up another phosphate, take it where it needs to go, drop it off. That's that energy transfer. So as it does this, once it becomes ATP, it's then like activated. And it's like, I'm energy, right? And then when it kicks that phosphate off again, you're releasing that energy. So now it kind of goes back to its lower state. It's like, all right, I'm just ADP. I'm not energy, but I can make energy. Then it picks up another phosphate. Boom, it's active again. It's adenosine triphosphate, ATP, our energy source. And then it's going to take it off and drop it off where it needs to go, becoming DP again. So again, it's from diphosphate to triphosphate. The triphosphate is the energy one. The diphosphate is almost like an enzyme. It's just kind of temporarily picking it up and then taking it where it needs to go and dropping it off. So it will get recycled. So the whole point of this is to make our ATP, right? So when we make our ATP, we have this energy stored in it. All we got to do is break that phosphate bond. We release the energy. Now the cell has the energy that it needs to actually perform all of its functions. So just like these enzymes that get reused, is so our ADP molecule. But what really is important about these enzymes is we know that they catalyze reactions, but we haven't learned about how they catalyze these reactions yet. And so this is what this picture is actually showing. So in this case, you can think of this molecule, this is known as the reactant because it's the one that's going to be reacting, right? It's the one that's going into the chemical reaction. Something's going to happen to the reactant. In this case, it looks like we're trying to break it up, right? So we're trying to break it up. So we have this this reactant that we need to turn into these products, okay? So we need to break up that molecule. Well, there's energy stored in that bond, which means it's going to take energy to break that bond. So we have, say, molecule 1 and molecule 2 right here. They're connected via this bond. This bond has a certain amount of, well, both these molecules have a certain amount of energy already in them. Already in them. Say they're little sugar molecules or something, right? They have energy in them. So that's what it means by here. They already have this level of energy. But they need to get over this barrier. This barrier is known as the activation energy barrier or the activation energy, which means you need to have this much energy minimum before the reaction will actually move forward. So we have this much energy, cool, we just need to raise it to that level. Okay, so what you would do in this case is you would just basically have to utilize your own body's energy and like force this molecule to break apart. That's going to cost you a lot of energy. So what your body does is it actually has these naturally occurring enzymes inside your body that will lower the activation energy. Now you don't need as much energy. You don't need to get all the way to here. You really just need to get all the way to here. So in this case, I already have this much energy. All I need to do is just add a little bit more. Not this big, this big amount right here. Just a little bit more. Just a little bit more energy and you can get the reaction going. Okay, so once you actually reach this threshold, then the reaction will move forward. So what enzymes do is they lower that threshold. So now I don't need to get all the way up here. I just need to get, to this level of energy. So I just have to add a little bit. So that's why it speeds up the reactions. Catalyzing the reactions by making them move faster because they've lowered the amount of energy that you need to make that reaction happen. So hopefully that kind of makes sense to you guys. If not, essentially again, the use of these enzymes lowers the energy required to get the reaction going. So in this case, we would have to add a lot more energy, which would take a lot more time. And therefore, the reaction would be slower than this one, right? Just a little bit of energy is needed. Therefore, this one will go much faster. And therefore, boom, the products will form with less energy required. So that's what these enzymes are doing. All right, so here is again that lock and key model that we kind of talked about already with these enzymes, but in greater detail. So we have our enzyme. Okay, in this case, it's sucrase. Now remember that anything that ends in ASE is probably used to chop something up. Okay, so sucrase, in this point, is the enzyme that's going to break up sucrose. Okay, sucrose, just like our lactose, right? The lactase, ASE, broke up the lactose. Sucrase breaks up sucrose. Okay, hopefully that kind of solidifies it a little bit. Because you will need to know this when we get to some of our labs. Make sure you know the differences between some of these enzymes and the substrate that they're trying to break down. So in this case, the enzyme is the sucrase, and the substrate that we're trying to break down, or the substance that we're trying to break down, is the sucrose. I know, too many words and I'm not looking at this. So, all right. So we have our enzyme. we have our active site. Remember, the enzyme is activated once the substrate, the proper substrate, fits into the active site. So that's what we have right here. We have our enzyme. We get the substrate. Oh, it's the right one. We're super happy. It fits right in here to the active site, so it binds to the enzyme. Now that enzyme is going to take it where it needs to go. So it's like, great, I'm ready for the chemical reaction. I'm going to take you where you need to go. In this case, we're going to add a little bit of water to help break up this molecules. Remember, that's the hydrolysis reaction, not the dehydration reaction where we're trying to build, right? This is the breaking. This is the hydrolysis, or cutting with water. Okay, so we add water, cut up those molecules, boom. Now we have the individual glucose and fructose that gets released that our body can readily digest, right? Sucrose is hard to digest. It's disaccharide, right? But these little monosaccharides, those break up really easily, so much easier to do. So the enzyme again would go pick up another substrate. It fits here in the active site, breaks up with the help of water, this hydrolysis reaction. We cut and we release the products. The enzyme is going to go pick up another substrate. Okay, so just like the ADP that we just talked about, this enzyme just is a temporary, well, it's a permanent molecule that is temporarily picking up something, dropping it off, going to get another one, temporarily picking up, dropping it off, going to get another one. So it's recycled. It's never used up. It's always just recycled. All right, now, sometimes your body's like, wait a minute, I don't need to produce any more glucose and fructose. Maybe I want to keep the sucrose. So in this case, your body would actually shut off the enzymes, and it can do this in a couple different ways. Now, one we can see right here is this is a normal substrate, which fits normally into the active site of this enzyme. So this is what it would look like when it's, doing its job. But now your body's like, you know what? Take a break. We don't need you to work anymore. We're good. So we're going to shut you off. So they can do this in two different ways. One, they can add an inhibitor as what's called a substrate imposter. So the substrate imposter pretends to be the substrate. So it's like, hey, I'm the substrate, even though it's totally not, but it fits in the active site just where the substrate would. So in this case, it is blocking or inhibiting the substrate. So now, because there's already somebody in his space, the substrate's like, well, I can't go here, so then he just bounces off and just goes on its merry way. Now, if we're not going to be using a substrate imposter, what you can do is you can turn off the enzyme another way by using this using a inhibitor that will actually change the shape of the enzyme. So in this case, we have this little inhibitor which doesn't even sit in the active site. It has nothing to do with the active site. But when this inhibitor attaches to the enzyme, it changes the shape of the active site, making it, in this case, too small. And therefore, the ball won't fit in it. And again, it bounces off. So the substrate would just go off because it can't fit in this active site, which means the enzyme essentially would be turned off. So here you basically block it by physically sitting in the spot where it normally would be. And this one, by sitting in a separate spot, you actually change the shape and therefore preventing that substrate from sitting in the enzyme. Same basic mechanism. They're both inhibitors. They're just kind of doing it in two different ways. That's all. All right, let's get into transport. This is like diffusion and osmosis stuff, hopefully, which you kind of remember from like elementary school and high school and stuff like that. And it is pretty straightforward and pretty simple. In fact, we're going to watch a video on it to kind of help solidify some of this information that's going on when it comes to diffusion and osmosis. So there is two different types of transport. One is known as passive transport, and it's passive because it requires absolutely no energy. He's just kind of like, whatever, do your thing. Now, this passive transport happens because of something called a concentration gradient, meaning you have a higher concentration on one side than you do on the other. When this happens, naturally occurring molecules, which are always just moving around, in liquid and in gas, these molecules are always moving around. In solids, they're like stuck. But in liquids and gases, they're always just kind of moving around. In fact, they're bouncing around. So naturally what happens is if you have a higher concentration on one side than you do on the other, these higher concentrations, as they bounce around, are naturally going to be able to move across that membrane until you have equal concentrations, also known as equilibrium. Okay? So we have a higher concentration here than we do here. These molecules are tiny. Anything in passive transport is probably a tiny molecule. Remember, it requires no energy. These large molecules are not just going to be able to pass through this membrane. Remember, the membrane is meant to keep things in and to keep things out. So little tiny molecules like water molecules and oxygen molecules, those are small enough that they can just float right through. Larger molecules will need some kind of help. So if it is a small molecule, right here it's a small and non-polar molecule, meaning it doesn't have a charge, then it can pass through the membrane, no problem. As long as you have a higher concentration on one side than the other, it's always going to go from a high concentration to a low concentration. Now, say you do have a molecule that's too big, or maybe it's polar, it's charged, and therefore can't pass through the membrane. In this case, you would use what's called facilitated diffusion. And that kind of helps facilitate or make diffusion happen. So the first type we learned about is just called diffusion or simple diffusion. This just naturally happens. These molecules bounce around until they move to equal concentration. Facilitated diffusion also goes from a high concentration to a low concentration until they reach that equilibrium, but they use kind of help, hence the facilitated part. Someone facilitated that and helped it go along. And in this case, we have a channel protein. One of those channel proteins that we learned about that are just kind of embedded in the protein, and it's just like an open door. Again, you have that wall, and someone kind of left the door open, so you can pass through that wall. Diffusion, you can think of, okay, I had a wall, but I'm a ghost, so I can go through the membrane kind of thing. I don't know. And then finally, we have osmosis. So diffusion and osmosis are still the same. They are going from a high concentration to a low concentration. When you're talking about diffusion, however, you're talking about the solutes. Remember, like the salt we learned about, or if I'm making chocolate milk, the chocolate, right? The little tiny particles, right? That's what's trying to move across the membrane. So my salt in this case would be trying to move across the membrane. But if I'm talking about osmosis, we still go from that high concentration to the low concentration. The only difference is when it comes to osmosis, we're talking about the water. For example, I know I would normally draw this on the board, but I don't have a board. Luckily, you can pause me and rewind me if this doesn't make sense to you. So, say we have a 10% water solution. Doesn't make any sense. 10% salt solution in water. So it is 10% salt, 90% water. Now, on the other side of our membrane, we have pure water. So it would be 100% fresh water, 0% salt. So in this case, the salt has a 10% concentration over here and a 0% concentration over here. So the salts are going to use simple diffusion. Where is it? Simple diffusion right here. And they're going to go from the high concentration, the 10%, to the low concentration, the 0%. So the salts are going to move this way. Okay, that's diffusion. Osmosis, you have 100% water concentration over here and only 90% water, because remember the other 10% is salt. Okay, so in this case, you're going to go from a high concentration to a low concentration, so the water is going to move this way. In this case, the salts and the water move in opposite direction. Okay, diffusion goes from high to low. 10% salt solution, 0% salt solution. Salts, diffusion goes this way. Water, 100% water over here, 90% water over here. High to a low, the water goes this way. Make sure you guys really understand that. It can kind of be difficult when you think high concentration to low concentration, but you have to be, what are you talking about? Are you talking about the water? Are you talking about the salt? Okay, so you always got to pay attention to which one you're talking about. Now, those are all passive transport, requires absolutely no energy. Always goes from a high concentration to a low concentration. However, sometimes your body's like, you know what? I don't want to naturally go from a high concentration to a low concentration anymore. I want to force things across the membrane. I want to create that gradient. Instead of that gradient just naturally being, your body's like, I want to make an artificial gradient. So in this case, I physically have to force things across the membrane in the wrong direction. This is called active transport. And active transport requires energy, hence active. You have to put in the energy. I'm forcing that molecule across the membrane. It doesn't want to go that way. It wants to go the other way. It wants to go from a high to a low, but I'm like, no. I'm going to keep moving it from a low to a high. So that's really important about active transportation. It's the only one that goes from a low concentration to a high concentration. Okay? Now, we're going to learn why that is in the next 2 lectures when it comes to cellular respiration and photosynthesis. But right now, you just need to know that it does, right? It goes from that low concentration to a high concentration because we are adding energy. We're forcing it across. So in this case, we have another one of those channel proteins, but in this case, it's a gate. So you have to go up and pay a fee to get through the gate. In this case, your fee is ATP. So you add a little bit of ATP to that gate. That gate's going to open up for you, allow your molecule forward. Okay, and that's going, again, from the low concentration to the high concentration. Unlike all these other ones, which are all going from a high concentration to a low concentration. So active transport goes in reverse. It's artificially creating a high concentration gradient over here. And again, we're going to learn about why that is in the next lecture. So don't worry about that today. Just remember, active transportation requires energy, is the opposite, and goes from a low to a high. Now, here's what we can see right here, the classic example of simple diffusion, right? We have a higher concentration on one side than we do on the other. So naturally, these molecules are going to bounce, back and forth, until they reach equilibrium. Now, this doesn't mean that for every molecule that goes this way, it stays that way. They're just bouncing around. They're always just moving around. So then, once they reach equilibrium, for every one molecule that goes to the right, you're going to have another molecule that goes to the left. Right? Maintaining that equilibrium. That's all that they're doing. Now, this does work with multiple solutes. So in this case, we have red on one side and purple on the other. Purple, excuse me. Purple is a high concentration on this side and a low concentration on this side. So purple's going to go to the right. Red is a high concentration on this side and a low concentration on this side. So red is going to go to the left. Okay, so they're both going to go separately. And again, one has nothing to do with the other, but they always go from a high concentration to a low concentration. So in this case, purple moves one way, red moves the other, until they both reach equilibrium on both sides. Okay, simple diffusion. Just the bouncing around of these little molecules until they reach equilibrium. And that's usually because these molecules want to be as far away from each other as possible. So here they're crowded, they're cramped, and they're like, why would we be so cramped? There's all this free space over there. Let's spread out until we all have equal space between us. That's why diffusion happens. That's all. That's all. Now remember that diffusion is, again, the movement of solutes, the little dissolved things like salt. And osmosis is the movement of water. Okay, like the water in the salt water would be osmosis. Sorry, that's the way. That would be confusing. Okay, osmosis, the water moving, diffusion, the solutes moving. That's all that is. All right, so with that said, let's go to our good friend Hank and watch some crash course videos. Guys, if you don't know about crash course, I think we've used them before in the past, but really super helpful. They have pretty much every single subject you can think of in college. I have all these really helpful videos. The guy talks really fast, just like I do. So luckily, again, you can just pause it. You can rewatch it. I'm going to embed this video in here. Hopefully it works. Usually Crash Course is okay with me putting their videos inside mine. Sometimes they don't. If they don't work, just type in Crash Course to the YouTube channel, and you can find all these videos. This one's called like Diffusion and Osmosis. So let's go ahead and watch that. All right, guys, thank you so much, Hank, for your wonderful Crash Course videos. I hope you guys liked it. If you weren't able to see the video in this video, go ahead and just go to the Crash Course YouTube site and you'll be able to find it. It's really just simple, just Crash Course, Diffusion, Osmosis. And like I said, they have pretty much all of them. You want to look up water, you want to look up carbon molecules, they have everything. They got biology, they got history. Really, really great resource, so go use it. I used it when I was in school, so really, really handy, guys. Go see it. I mean, if you haven't just watched it, because it worked. Hopefully it works in my video. All right. So, we learned a little bit about this in the video, hypertonic, hypotonic, remember hypertonic and hypotonic, right? So a hypertonic solution has a lot of something, like a lot of energy. Hypotonic solution doesn't have a lot, it's got a little bit low energy you can kind of think of. And we're just talking about solutes, so high concentration of solutes, low concentration of solutes. If you have the same, meaning you are at equilibrium, this is known as isotonic, or the same. So hypertonic, hypotonic, isotonic, the same, equal concentrations. Now, it is crucial that some of our cells be hypertonic, isotonic, or hypotonic. And you're like, wait, really? Yes. So cells need to maintain certain water concentrations. And if the solutes kind of get out of the, remember solutes go one way and water goes the other. So if this happens, we can actually have dehydrated cells or cells that are overhydrated and can be bad. So let's look at the top first. So ignore the bottom. We're going to be just looking at the top. We're going to be looking at red blood cells because we're animals, right? These are animal cells. Notice because they're big, round, and squishy, unlike these little geometric plant shells right here. So we have an isotonic solution, meaning our blood, the little red blood cell, and our body are the same water concentrations. If they are the same water concentration, for every water molecule that comes into my blood, a water molecule is going to leave. Right? Maintains a nice, happy, healthy blood cell. That's okay. However, if I were hypotonic, now remember it's very important you pay attention to the wording of these. I'm talking about a hypotonic solution, meaning the stuff on the outside is hypotonic. Okay, so the stuff on the outside has a lower concentration of whatever solute we're talking about. Let's say in this case salt, just as an example. So there's less salt out here than there is in here. So in this case, the salts are going to leave the cell. Remember, because we had a lower concentration here, hypotonic, versus the inside. So this would be hypertonic, this would be hypotonic. So it's going to go from a high concentration to a low concentration. If the salts are coming out, the water is going to rush in. Remember, they go in opposite directions. Salt goes out, water rushes in. Now your little red blood cells like, and it's actually going to get way too much water rushing in, and it's going to cause the cell to lyse or burst. Not good. You don't want your red blood cells bursting, right? That's a bad thing. Alternative, say we have a hypertonic solution. Now there's a lot of salt solutions out here, hence the dark blue color. We have a lot of salt out here. We have very little salt out in here, which means the salts are going to go in, which means the water is going to come out. Okay, so now the water comes out, and now you get like an emaciated little cell, also not a happy cell. So you don't want a hypertonic solution. You don't want a hypotonic solution if you're an animal cell. You want an isotonic solution. So you want our blood and our internal environment to be roughly the same concentration. Now, if you're a plant cell, this actually works a little bit different. So if you're a plant cell and you have a isotonic, or you're in an isotonic solution, for every water molecule that comes in you, one's going to leave. But wait a minute, you're a plant cell. You need water. You need lots of water. So in this case, you can never get enough water that you need. So in this case, you're actually kind of shriveled. So you're shriveled or wilted. Like, how plants like, I'm happy. You don't water it enough and it gets sad and starts to wilt. That would be if it's just isotonic with the outside environment. Not good. Also not good would be the hypertonic solution. So now you have too much salt on the outside, which means salt's going to rush in, which means your water's going to rush out. Now you never get enough salt. So in this case, you're still shriveled. So in this one, you're flaccid. You're not getting the pressure that you need to bulk up and be nice and happy. You're just kind of sad. And in this case, you're shriveling. You're really dying. This is where your plant dries up and essentially just and dies. Bad. So you don't want to be isotonic. You don't want to be hypertonic. In this case, you want to be hypotonic. Okay? So on the outside, the salts are low, which means the salts are going to flood out of the cells to the environment. But what's going to flood in? Water. Now, as a plant cell, you have all the water that you need, and now you're super happy. So very important for these plants to be in a hypotonic solution, meaning less salt around them, or whatever solute you're talking about. And I just use salt as an example. So now you're going to have water constantly flooding in, which for you as a plant cell is going to be a good thing. So again, we have a hypertonic, isotonic, or hypotonic solution, right? Hypotonic solution, happy little plant. Sad, wilty plants would be, your isotonic or your hypertonic solutions. Not happy plant. Keep your plants happy. We need them for oxygen. All right, so remember we saw this in the video. We kind of talked about this a little bit already. Active transportation actively requires energy, right? You have a channel protein, but it's closed and it requires a phi. That phi is energy. Now you add the little ATP, boom, it opens up. Now you can actually move stuff across the membrane. And remember, when it comes to active transportation, we're always moving it in the opposite direction, right? We're moving it against the gradient. So that's why we're doing active transportation, because it wants to go from a high to a low, and we're like, nope, you got to go from a low to a high. But that's going to cost me, and I'm okay with that. I'm just going to add some ATP, push that across the membrane. So again, we have a lower concentration over here, and we're moving to a high concentration. We're shoving them across the membrane to keep them over here to create this concentration gradient. And remember, we're going to need that next week when we start talking about cellular respiration and photosynthesis. All right, so we've kind of learned about this already when we were talking about the cell membranes, but sometimes your body needs to get rid of stuff out of its cells. And remember that these little lysosomes or transport vesicles or stuff like that, they're all made of the same type of membrane. So say we needed to get rid of something out of our cell, and it's like, all right, too big to go through the actual membrane itself. I don't want to require too much energy, so I'm not going to bother with active transportation. We're just going to send it outside of the cell via these vesicles. So that's what this is right here. This is a little transport vesicle, and it's taking a protein that we just made and making it leave the cell. So inside we have our little protein molecules. Inside our transport vesicle, that vesicle is going to come up to the outside of the cell membrane, It's going to attach to the cell membrane because they're all just phospholipids, right? And then it's going to open up, releasing these molecules, whatever they are, and to the outside of the cell. So we're on the inside. Now we're moving on the outside. This is known as exocytosis, or exiting the cell. Right? Pretty straightforward and simple. Now, play that exact thing in reverse. We have endocytosis. Say we need to bring things into the cell. We're like, all right, we got these proteins or salts or whatever they are out here. We need to bring them in. So essentially all the membrane is going to do is it's going to lay flat, and then it's going to start to fold in on itself, creating a transport vesicle. And that's what you see right here. So it starts to pinch, trapping whatever solutes we want on the inside and bringing it into the cell. This is known as endocytosis, or entering the cell, coming into the inside of the cell, hence endo. Okay, so exocytosis, we are leaving the cell, transfer vesicle comes, open up, releases it outside of the cell. Endocytosis, the exact opposite, closing up, bringing into the cell, endocytosis. The last one we have, actually there's a couple different forms of endocytosis. One of them is known as phagocytosis or phagocytosis, pronounced both ways. This essentially means cell eating. So say you're a cell, like an amoeba, right, that needs energy. It's going to go out and it's going to eat a molecule. And that's exactly what we can see right here. So this would be one of those pseudopods in that amoeba that we saw in that video. Hopefully you watched it. And that's all they're going to do. They're going to extend their cell membrane around the food particle or whatever it's trying to eat. They're going to close it up and they're going to bring it to the inside. Endocytosis. But in this case, we're bringing in food. Hence, phagocytosis. Now, there's another one called penocytosis, which is essentially cell drinking. So phagocytosis is cell eating. Penocytosis is cell drinking. Say you have some nutrients out here that you need, your cell needs. And it's like, all right, I'm going to do the same thing. I'm going to start pinching off. I'm going to bring in some of that liquid, and then I'm going to bring it and drink it up. Hence, penocytosis, cell drinking. And the last one is receptor-mediated endocytosis. And so essentially, it's only going to be triggered by those little receptor proteins that are embedded. So only when the signal comes and sits here are you actually going to have that endocytosis. So we have these little receptors. They receive the signal. Now they're going to close off, pinch off, and start bringing in these little whatever was received by the receptors. Sometimes it's like a quantity issue, like there's not a lot of them. So we're going to wait until we actually grab one, then we're going to bring that in, that kind of thing. But it depends on the type of endocytosis that your cell is doing. So it varies. All right, with that, we have some passive humor right here. Get it? Passive. Anyway, thank you again for another wonderful lecture. I really appreciate you guys sticking around with me. And I really hope that all of this information is kind of being solidified in a helpful way. So if you have any questions, comments, anything, of course, make sure to email me and contact me. And I will just see you next week for cellular respiration.